Hello all. Within the frame of the Malagasy Grassy Biome Workshop, I would like to present a brief overview of how can genetic data be used to infer landscape changes over time, and therefore how it can be used to somehow inform us about the expansion of grassy biomes. The underlying idea is that for a particular landscape, we can infer processes of forest loss, forest fragmentation, and open habitat expansion using the genetic diversity of forest-dwelling species, such as mammals or trees, and of open habitat species, such as grasses or shrubs. From genetic or genomic data, we can infer the demographic dynamics of the target species, which dynamic can be used as a proxy to assess landscape changes. And here you have a plot of the PSMC conducted by Elena Teixeira that she presents in her own talk, and it's one of the classical demographic inference that you can obtain from full genome data. Many approaches and methods have been developed since the early 90s to infer demography over time, but describing these methods is not the purpose of the meeting and is covered elsewhere. In Madagascar, this kind of approach has been early used by the team of Ute Rattespiel and Rune Schicki, for example, to assess the population trends of many lemurs across Madagascar. For instance, Olivieri and Collet, in their work published in 2008, inferred a strong and concomitant population decline in 3 neighboring mouse lemur species of northwestern Madagascar. These declines across all sites and species were found more likely to have occurred in the past 500 years. This suggests that the northwestern deciduous forest, the habitat of these 3 species, may have suffered a major decline within this time frame. In northern Madagascar, Lunes's group combined forest cover change monitoring from remote sensing data with population genetic demography inferences across the fragmented forest distribution of the golden-crowned sifaka Propithecus tattersallii. Their analysis showed that the forest cover did not change dramatically since the 1950s, a situation contrasting with the rapid deforestation recorded elsewhere in Madagascar. Furthermore, they inferred a strong population decline of the Tattersallii sifaka that they could date to approximately 10,000 years before present, and with a high probability to have occurred before the first traces of human arrival known at the time for Madagascar. The same data was later reanalyzed together with the neighboring Propithecus perieri data and using panels of models mimicking habitat loss and or habitat fragmentation, in other words, considering both the processes of population decline and population structure. This study revealed that both neighboring species underwent a strong co-occurring bottleneck that was accompanied with habitat fragmentation. In contrast with the study of Kemmerer, the dating of this event was more recent and coincided with the mid-Holocene transition as well as the first human traces in Madagascar. In the same region, a recent study from Guillaume Bénard's group revealed surprisingly strong correlation of the genomic diversity of the spiny Malagasy olive forest tree with the forest cover of the Lokimanambato region. This correlation, considering the long generation time of this tree and the time necessary for such strong pattern to emerge suggests that the shape of the forest patches in the Loki Malambata region has been relatively stable for a long time, easily 1,000 years. This signal seeks for deeper analysis of the spiny Malagasy olive demographic history to better assess the time of the forest fragmentation in the region. Similarly, the analysis recently done by the group of Anyoder in comparing two species of mouse lemur in central Madagascar and western Madagascar showed a higher genetic distance across population of Microcebus leila, suggesting that these populations long lived in a mosaic environment. In line with the work of Jan Hakel on grasses that is presented in his own talk, the group of Guillaume Bénard also revealed that the diversification of the only savanna-dwelling Malagasy olive tree, Neuronia lori, coincide coincide with one of the major savanna expansions across the globe around 6 million years in the past and shows in a few occasions the occurrence of clonal growth, a potential adaptation to fire-prone environments. In brief, this genetic-based inference of past landscape dynamics have a lot to tell us, but only if we keep in mind their limitations and caveats. That is, first, all these methods are based on models, which are only relevant if they are close enough to real demographic history of the studied taxa. For instance, the team of Lunasiki has been extensively working on the effect of structure on demographic inferences, and their most recent work demonstrates how one can interpret a PSMC plot as a fluctuation of the population size through time, which is the most common interpretation of a PSMC plot, or as a metapopulation of constant size, but with changing connectivity through time, as shown here by the vertical lines. In short, one needs to have a proper understanding of the species biology, ecology, and structure to develop models realistic enough to hope for an accurate inference of its demographic history. Second, the dating of demographic events are scaled by mutation rate and generation time. Estimates of these parameters are not always available, and estimating accurately these parameters can be challenging. For instance, in mouse lemurs, several values of oversimplistic generation time have been used until a couple of more holistic and accurate values were proposed recently by Udo Huddersfield and Ann. The most recent value were used in the work presented by Elena Teixeira and Vincent Monda in their talk about the Amber Mountain vegetation history. And this calls for the re-evaluation of previous results with the new most accurate mutation rates and generation time. To conclude on the potential contribution of population genetics demographic history studies to the understanding of Malagasy grassy biomes, I believe that its potential lies on the use of comparative studies that will include multiple species from forest and open habitat environment from studied regions. In particular, no demographic history study has been conducted yet on Malagasy grasses. And these, the Malagasy grasses, they hold a great potential to inform us on the past anatomy of Malagasy grassy biomes, whether it is by reconstructing invasive or endemic grasses expansion as well as forest decline. However, they challenge our way to assess the demographic history from genetic data because of their polyploidy in many cases. Furthermore, our ability to interpret the results of demographic inferences relies on the availability of existing local sources of other type of data, like forest cover reconstruction or paleoenvironmental data. Therefore, population genetics definitely benefits from cross-disciplinary study. This was a brief overview of how genetic data has been used to inform us about the expansion of grassy biomes and how it can be used. Thank you all.